Uses of science of heredity. Number 1, diagnosis, treatment and prevention of hereditary disorders. Number 2, production of hybrid varieties of plants and animals. Number 3, industrial processes involving microbes. Genetics is a branch of biology concerned with the study of genes, genetic variation, and heredity in organisms. Though heredity had been observed for a period of 1,000 years, George Mendel, a scientist and a friar, meaning a monk, working in the 19th century, was the first to study genetics scientifically. Heredity is used in pedigree analysis, a method that diagnoses the presence of genetically transmitted diseases. It is helpful to avoid the expression of the disease in the descendants. Genetic research is creating new ways for people to take action and prevent disease, and new ways to treat disease through personalized medicine. That is, individuals based on unique genetic makeup. For example, genetic disorder associated with heart defect might be treated with surgery to repair the defect or with a heart transplant. Conditions that are characterized by defective blood cells formation, such as sickle cell diseases, can sometimes be treated with a bone marrow transplant. Sickle cell diseases is a group of disorders that cause red blood cells to become misshapen and break down. Sickle cell diseases is an inherited group of disorders that affects hemoglobin in red blood cells that delivers oxygen to cells throughout the body. The red blood cells become sickle-shaped or crescent moon-shaped. The cells die early, leaving a shortage of healthy red blood cells and can block blood flow, causing pain. Bone marrow is a spongy tissue inside some of our bones, such as our hip and thigh bones. It contains stem cells. The stem cells can develop into red blood cells that carry oxygen throughout our body, the white blood cells that fight infections, and blood platelets that help with blood clotting. Bone marrow produces 200 billion new red blood cells every day. Genetic disorders are diagnosed through specific tests, which can include examining chromosomes or DNA, that is, the proteins that make up genes, or testing the blood for certain enzymes that may be abnormal. Studying enzymes is called biochemical genetic testing. Genetic disease prevention and treatment check regularly for the disease, follow a healthy diet, get regular exercise, avoid smoking, tobacco, and too much alcohol, get specific genetic testing that can help with diagnosis and treatment. A gene is the basic physical and functional unit of heredity. Genes are made up of DNA. Some genes act as instructions to make molecules called proteins. However, many genes do not code for proteins. In humans, genes vary in size from a few hundred DNA bases to more than 200 million bases. A unit of DNA has 4 bases: adenine, guanine, thymine, and cytosine. Bases are the part of DNA that stores information and gives DNA the ability to encode phenotype, a person's visible traits. Adenine and guanine are purine bases. These are structures composed of a 5-sided and 6-sided ring. Cytosine and thymine are pyrimidines, which are structures composed of a single 6-sided ring. Mutations is a change in the DNA sequence. Mutation can result from DNA copying mistakes made during cell divisions, exposure to ionizing radiation, exposure to chemicals called mutagens, or infection by viruses. Can mutations ever be advantageous? Mutations can be beneficial, benign, or malignant, depending on where in the genetic code they are located. Examples of beneficial mutations include HIV resistance, lactose tolerance, and trichromatic vision. Beneficial mutations lead to new version of proteins that help organisms adapt to changes in their environment. Beneficial mutations are essential for evolution to occur. They increase an organism's chances of surviving or reproducing, so they are likely to become more common over time. In short, a harmful mutation decreases the fitness of the organism, whereas a beneficial mutation increases the fitness of an organism.